Okay, welcome to microscopy. Um, Hopefully you guys have gone through the little, uh, tools of the lab and some lab safety. I didn't record anything because you can read that for yourself, and I have little YouTubes for you guys to watch. But I did want to go through microscopy and staining and classification with you. Hopefully, this will go along with some of the prokaryotic cell lecture that I've done, and you can take a look at that. But microscopy is very important in obviously, because it's one of the few ways that we can look at these guys. We can use an electron microscope, but trying to use a microscope is how we look at these bacteria. And when we talk about microscopy, we have to talk about some general principles, looking at wavelength, looking at magnification, resolution, contrast. I know you're not in the lab, so I'm trying to get you through this. This is about the best I can do here. We're utilizing the electromagnetic spectrum. Okay, we are using visible light, and it does make a difference. Resolving power. The resolving power increases, and we'll talk about this, as you get into the shorter wavelengths. Just FYI. So the shorter wavelengths we get in, the better we can see these guys. One of the things that we use is, obviously, we are using lenses. So we are refracting light and we are magnifying, and that becomes very important. If you were in a lab with me, we would talk about it. Basically, we have these lenses, and when you use a microscope slide and a cover slip, they all have to be the same refractive index so that everything refracts together. If you have something that's of two different refractive indexes, you can't see anything. You have to bend the light all the same way. Okay, so that becomes very, very, very important. And so we're going to have our specimen, we're going to have our lens, and we're going to have a focal point that allows us to see. But we want this light to bend all in the same way. If it's all over the place, we can't see anything. Okay. The limits of what we can see are here. Unaided eye, the large protozoans, you can see some of them. And you get into below that, we need a microscope. We need a microscope to see red blood cells. We need a microscope to see chloroplasts in a leaf. Hopefully you guys have seen that in some biology lab, right? A typical bacteria and archaea, we can use that microscope. And we use that compound microscope. We can see mitochondria. Now we're getting down towards the bottom. We're getting back down there towards the bottom. Anything below that, when looking at pieces and parts of these organisms or viruses, we need an electron microscope or a tunneling microscope. These are pretty expensive. Luckily, where I worked in Florida, I was able to see both a transmission electron microscope and a scanning. They are large. They take up an entire room. A lot of times, at least an entire desk. We got pictures. But understand that once you get below, when you're trying to look at organelles or something like that, or the ribosomes of it, you got to have an electron microscope. But the bacteria we can see, we can stain, and we can, we're able to visualize them. Okay. Couple concepts that we need to go through is contrast and resolution. I mentioned it. Using blue light helps in the resolution. Contrast is the ability to distinguish two objects as two, the difference in the intensity. Can you see these objects from the background? It's the ability to determine this organism is separate from the background that you're looking at. Okay, so the way we do this is we stain. Okay, Staining increases this contrast, and I'll show you a picture with that. If you're trying to look at bacteria and it's in a broth or something like that, you can barely see them. Now we'll do motility, we can see them move. Oh, we really can't see very much. But if you put a stain on it, that's a different story. So we can use light. We can also use a phase scope. We'll talk about phase, but we can put this in and out of phase to increase the contrast. Okay. So here's an example of contrast. You can see this is unstained. You can see these lovely little bacteria. They're bacilli, guys. Yeah. And we take that same stain and we slide and we stain it. And that contrast is much better. We can see a lot of that, the outside of that bacteria. It allows us to get the outside shape, okay, and size. And I try and tell people, when you use a single stain, we'll talk about this, a simple stain, there's a difference in these stains. But when you use a simple stain, you can see the size and you can see the shape. But that's it. You can't tell anything else beside that. All right. But you can at least be able to see the size and shape of the bacteria versus, it's kind of hard. All right. So we do use these light microscopes. We have bright field, dark field, phase, fluorescent, confocal microscopes. Yep. All kinds of, most colleges will have at least bright field, dark field, and phase. They may not have fluorescent scopes. A lot of hospitals will have fluorescent scopes. I've worked under the fluorescent scopes many times. Okay. So bright field, two different kinds. Simple microscope, compound. A simple magnifying glass is a simple microscope. I mean, you can take a magnifying lens and look at something. What Leeuwenhoek did was use a simple microscope to observe these microorganisms. Well, this is great and good and dandy, but it doesn't help us. We need more magnification. So the standard microscope that we use is a bright field compound microscope, and it uses a series of lenses for magnification. And the light rays pass through the specimen and into the objective lens, because the light is going to come up from the bottom through that specimen and into the lenses. A lot of times we will use oil immersion lenses. If you look at these microscopes, they usually have 4 little heads and they are different magnifications. All right, 4x, 10x, 40x, and oil are the standard ones. Okay. The 100x is an oil immersion lens, and oil immersion means it has to be in oil. So there will be a drop of oil placed on the slide, and then the oil immersion lens will move into that, and that gives you your 100x magnification. Usually you have 1 or 2 ocular lenses, your eyepiece, the lenses in your eyepieces. Most microscopes that we use are binocular. Okay. So the total magnification is the magnification of the objective lens times the magnification of the ocular lenses. Usually your ocular lenses are 10 times, 20 times, but usually 10 times. And so if you've got an oil immersion lens at 100x and you've got a 10x ocular, you have a magnification of 1,000 times, which is what we normally see in an oil immersion. Okay, and if you're using what we call, you have high power and low power. A high power lens is usually 40x. That would be a 400 times magnification. Okay, just FYI. So there we go. So there you can see this one has 4. There's one over there. The longest one is always the oil immersion lens. So the short lens very low power, 10x, and then you go up, and the longer the lens, the higher the power it is, okay? And you can see this is a binocular, and you can actually, with this particular one, you can put a camera on it. But there are the light sources with a lens coming up through, going through a lens, going through another lens. So this is how we get the magnification. We do have dark field scopes. This is best for observing very pale objects so that you can see, and external features. If you're in a real live lab, what we do is usually put like live organisms down, like a bacteria, and you can actually see a dark field. A bright field you can't see too much unless you stain it, and if you stain it, the organism is dead. You have to kill the organism to stain it. What dark field allows us to do is look at this bacteria while it's still alive. So we can see flagella, we can see motility, we can see fimbriae, we already talked about that. So we can see these external features. It's really good for the external features because they're being lit, all right? And you only get the light rays that are scattered by the specimen entering. The specimen appears on a dark background, okay, and it increases the contrast. This is the dark field lens that fits, we'll go back here, right over this condenser. It fits right in there. It looks like that. You just slip it on and it goes right in there, okay? And what happens is the only light coming through is the light that's coming through on this outside ring. Everything in the center is stopped. So the light unrefracted from the gets through, okay? This is dark field. This is what it looks like. And you can see, you don't really see a lot of features on this image. Can you see the edges and stuff like that? And that's what dark field, we really use dark field for. All right, phase is interesting. We can look at this. We use a set of light rays that are two sets. One is a half wavelength out of phase. And what a phase scope is metaphor with, when we talked about dark field, we said it's the external features. Phase scopes is the internal features. So you can go back and forth with these different microscopes. And I will tell you, in most college labs that I've worked in, you had these microscopes that we could easily convert from dark field to phase, a piece of cake, okay? So you have, if you go back to the microscope, this would be a normal bright field. You put that filter on and here, and it becomes dark field. There's usually a little phase button, and if you push it in, it's then a phase scope. It kind of looks like this dark field filter is just placed in a different place in the line of sight, okay? So what happens is we're all in phase, and the rays are out of phase. Peaks and valleys are different, okay? What you do is, here's your bacteria, here's the phase plate. Again, it looks a lot like the dark field, but the light rays are coming in, and when they come in, can you see they're half a wavelength out of phase, usually, and they hit the phase plate, and they're now a quarter wavelength out of phase from the surrounding. So if you just remember that and try and get that picture, that's what you get, okay? And it will make it a lot easier. So here is an example of what it looks like. You can see bright field, and I can't really see the edges. I can see the general features. Dark field, you got a dark background, and you can see a lot more of this outer edge makes it a lot clearer. Phase, look at that. You get a lot of differentiation in the middle and stuff like that. So you can really use these to, I mean, this is a lot clearer than that. Okay, so we use this a lot in micro. We can use fluorescence. We can put a fluorescent tag and have these glow. We used to do that way back when to test for syphilis, and we'd attach a fluorescent tag to the spirochete, and if we saw little spirochetes, we could diagnose it. But basically, we take UV light as the source, and it causes the specimen to radiate energy back. It does increase resolution and contrast. We'll talk about a resolution in a second, but some cells are naturally fluorescent. Some have to be stained with a fluorescent dye. Okay, we can also use this is common in the laboratory, is using immunofluorescence, where you attach the fluorescence to an antibody to identify the pathogen. Okay, and here's our little fluorescent scope. These are a little bit bigger. Okay, and you can see the little detector. But what you do is you tag it. Here's your antibodies. You take the fluorescent dye, you tag it with the antibodies. The antibody gets attached to the bacterial cell, and boom, we glow. Okay. You got to remember, if you have a fluorescent microscope, you got to be in a dark room. It doesn't have to be in a light room. It does not help at all. Okay. And the one thing we have to talk about is resolution. I talked about the ability to see resolution. Well, resolution is the ability to see two objects as two separate and distinct objects. So as you go up in these microscopes, the ability to see these objects increases. You know, when you get there, I can definitely see this is separate from this, this, and this. As we go up in magnification, that ability to differentiate, that ability to resolve that these bacteria are two separate bacteria become important. Okay, does that make sense? Very rarely would we use an electron microscope. I mean, this is magnifications up to 100,000x. But if we're going to look at a virus, if we're going to look at internal structures, we have to do that. And there is a slight difference. This is the TEM. It's a very big unit. It is a very tall unit. I mean, it doesn't look at that, but this thing is about 10 feet tall at least. I mean, and this is what you get. Okay. Your SEMs, your scanning electron, very good at looking at that outside. Look at that. Beautiful. Okay. Now, we're going to briefly go into stains, because in order to see these things, in order to do microscopy in bacteria, you got to stain. And we're going to go through stains in detail in the labs. But it allows us to increase the contrast and the resolution by coloring these specimens. Okay? So we take a thin smear of these organisms, apply them to a slide, then we fix it. We have to heat it. These stains are water-based, and I don't know about you, but you put water on a slide, it's going to wash off. So we literally have to, quote unquote, fix these organisms to the slide, which means we pass them. You'll see it in a little video I have. You pass it very gently, quickly through heat, through a flame in a Bunsen burner, or apply heat, and that adheres that bacteria to the surface of the slide and allows us to work with it. Then we go in with a stain. Okay, it usually has either cation salt and is colored, and we can have acidic dyes and we have basic dyes, and this does make a difference, and we'll talk about this in a little bit later. But there are several different stains that we use: simple stains, differential stains, special stains, and then staining for electron microbes. Okay. The simple stain, as I mentioned, is only one simple thing. It is one color. That's all we can look at. And when you look at a simple stain, yes, I can see cocci, and I can see them singly. I can see them in doubles right there where the arrow is pointing. I can see them clustering around. This is very typical of staph, but I really can't call it a staph. It is a staphylococci in that it is clustering. But I can't really say anything else because I've only stained it with crystal violet. And as I said, when here's it unstained, same thing. You put it in a stain, you can see it. I can see the size and I can see the shape. That's all I can give you at this point in time. Okay. And what we do in general labs is stain them with different colors so that you can see the difference. There are several different stains we use in the micro lab. And so you can stain with blue, you can stain with green, you can stain with purple. We've got all kinds of fuchsia that you can stain these guys with. Okay. Then you can have a differential stain. That means you can tell the difference in something. Okay. Gram stain, okay, is more than one color. And there is a procedure. You lay one color down, you decolorize, and then you lay another color down. This is a gram stain. We'll talk about this. But here you can differentiate. And remember we talked about in prokaryotes, we talked about the difference in those cell walls, okay? So we'll go into the gram stain in another lab, but here you can see the gram-positive cocci, these dark purple cocci, okay? And gram-negative bacilli. You see that guy? All right. So do you see that? And that's what a gram stain allows us to do. Differentiate whether this organism is a gram-positive or gram-negative and the shape of the organism. And this is why gram stain becomes so important because it's a lot of information to the doc. All right. And that is our very typical, most common stain that's done in a micro lab. Okay. Another one that's done is acid fast stain. This we are looking for primarily tuberculosis, or maybe looking for leprosy, but primarily it's tuberculosis. We're getting down sputums, and we're looking for acid-fast bacilli because that's what tuberculosis is, and we'll talk about it. And this is, again, it's a two-color stain. It's a special way of doing it, and you can see these acid-fast bacilli against a staph that we use as a control sometimes. Okay, but again, two different colors, and we can tell the shapes. So a lot more information on these differential stains. We can do an endospore stain, and we'll talk about that when we get to that lab. But there's your little endospore in there. This allows us to, this is obviously a member of Clostridium or Bacilli because they have endospores. There are times when we want to know, is it forming endospores at that time? Okay. Is the environment bad or is the environment good? Because if the environment's good, it's not going to be doing endospores. All right. We can also do these special stains, negative stains, flagellar stains, fluorescent stains. Okay. I'm back here. This becomes important. We use a negative stain. We do this. We'll talk about this. India ink preps. It's a dark, dark, dark stain that's done on spinal fluids. We're looking for a particular organism that can cause encephalitis. But what a negative stain allows us to is basically to learn that the bacteria have a slight negative charge because of the cell walls. And the stains that we mostly use, your gram stains, acid-fast stains, have a positive charge. so that the negative and positive charges attract. And it's attracted to that cell wall, it pulls it in, and it holds it. A negative stain is negatively charged. So negative repels a negative. So you're using a negative stain to look at this. And this is a real quick way to look for something. Again, any ink prep that we do, we're looking for Cryptococcus. And, it takes 2 seconds to put India ink on a dot of spinal fluid, put it under a microscope. Yeah, I can see something. No, I can't. That quick. All right. Flagella stain. Maybe we want to look at the flagella. Whoop, there we go. There's the flagella. So hopefully this gives you a little introduction to microscopy and staining. There's going to be some YouTube videos that I'm going to want you to watch. And I'll also be going through some of these other ones. But hopefully you will have a little bit better feel. And this is about as best as I can do to get you into the laboratory. Okay.